So one of the key things we'd like to know that relates to specificity is what's the NPS really measuring? On the one hand, it could be something very specific. Maybe it's only thermal pain on the left arm, or maybe it's any kind of somatic pain on the body, or maybe it's just anything that's negative and emotional, affective, or maybe anything arousing, or just anything salient altogether. All of these things tend to increase as pain increases. And another way of asking this question is, what is our NPS signature really representing? What's it capturing? One way to think about this is in terms of psychological receptive fields. So any receptive field has a set of things that activate it and a set of things that don't activate it. And we see those things inside and outside the circle, respectively. So over here on the left side, here's an example of a PRF for something that responds to negative emotional things. It would be activated by noxious heat, noxious pressure, rejection, anger induction, aversive images, vicarious pain, watching other people in pain, all these things. But it wouldn't be activated by things that are not emotional, like light touch or doing math problems, arguably. At least not for Martin. On the right, this is a PRF for something that really responds to somatic pain. It's activated by multiple types of pain, heat and pressure, but not by any of these other negative arousing things. And one way to examine this is by examining social pain, rejection. So social pain hurts. We often use the same words, and many people think that this is more than just a metaphor, that these threats to our social well-being are actually partly mediated by the same physical system that processes painful somatic events. So in this next study, we had 60 participants. All of them were romantically rejected. They all reported not being over it, and they were the recipient of an unwanted breakup. So what they did is they brought in pictures of their ex-partners and a close friend, and then they also experienced painful and non-painful heat. So the design is very simple. We have equal intervals of all four of those conditions. And what's interesting about rejection and pain is it produces similar ratings of negative emotion and similar patterns of brain activity. In fact, if we look across regions, at regions that are activated both by physical pain and by rejection, then they look really similar. And there's nothing that we've seen that's actually more similar in terms of the pattern of brain activity than looking at the face of the person who rejected you. So there's common activation in the anterior cingulate, the anterior insula, and also in the secondary somatosensory cortex and dorsal posterior insula, which is otherwise activated relatively specifically or quite specifically by somatic events, things that happen to your body. So this basic similarity provides a really nice way of testing our pain signature and whether it's really specific to painful events. We can first ask whether it picks out, when it's somatic pain, which is the more painful event. And yes, indeed, in 90% of the sample here, it successfully tracks which is the more painful somatic event. Now, what about the rejectors? Is it going to activate our pain signature? Well, it turns out it's not different from chance. So there might be a little bit of a response there, but then when we look on the right at the expression across the different conditions, we see that for the high pain condition, there's a strong response. For the low pain or non-painful warmth condition, the response is below threshold, which is the dotted line from the previous studies. And then the rejector and friend photos really don't activate at all. So in this case, we can conclude that it's really specific to physical pain, at least in this case. And this allows us to start to look at the patterns themselves and compare their similarity. So the anterior cingulate is supposed to represent the common basis for negative affect. And what we can do then, we can look specifically in the anterior cingulate, and we can ask, can we identify a pattern that tracks the pain itself and what happens and doesn't respond to the rejection? And indeed we can. We can identify a pattern in the dorsal anterior cingulate that is significantly responsive to the pain, but it's at chance for the rejection. Then we can try to identify a pattern that's similar for rejection. We can identify a rejection-related pattern that's sensitive to rejection, but it's a chance for physical pain. Now this double dissociation, it's also called separate modifiability, is a strong constraint on the separability of those patterns and the underlying representations in dorsal anterior cingulate. The patterns are uncorrelated and each is separately modified by a different intervention. So in this case, we don't see any evidence for shared representation in pain affect regions. Rather, we find evidence that there are different patterns that represent different kinds of affective processes, all within the dorsal anterior cingulate region. And when we test whole brain patterns, they're also separately modifiable in very similar ways. So this can help us infer something about the nature of the underlying representations involved. And finally, I will speak to the idea that we need to take the best results and promising markers from all areas of neuroscience, and we need to take the best and we need to ramp them up. We need to share them and test them across laboratories and conditions. So as we started to see here, really developing and testing the specificity of a marker like the NPS requires testing across multiple studies, and it's going to require larger and larger samples and more diverse populations in order for us to have confidence that it's going to work for new individuals in a very large, broad population. So the idea here is from David Borsick's work and others in the Biomarker Definitions Working Group. And the idea is that the biomarker development pathway parallels similar developments in clinical trials. And you start with discovery of a biomarker, an initial validation and demonstration, and as you proceed and things look promising, then you can characterize it more completely and finally start to use it in very broad populations and evaluate its suitability as a substitute or as an adjuvant for other measures. And as you move up this ramp, it requires more and more effort and larger expenditures. But we should, as a field, take the most promising of these markers and results and tasks and scale those up gradually as they pan out in the literature and become useful. So this is the idea of turning a brain finding into a research product that we can share across laboratories. At this point, we've tested the NPS, the pain signature, across a number of different research sites across the world, and we're very interested in testing it on all kinds of conditions and all kinds of things that might be confusable for pain, that might challenge or define its boundary conditions or help support how it generalizes to new populations. So this is one view of the step towards this broader dissemination and development that's going to be very important for our field. So that's the end of this module. Thanks for listening.